All right. So, welcome. This is part of the video that's associated with Lesson 12 on drugs of abuse. So, since we use mice to uncover a lot of the neurobiological underpinnings of how drug abuse and the effects of drugs and addiction work, it seems fitting that we would go inside of a mouse party that simulates taking different types of drugs of abuse. These mice are doing it, as you can see, and we're going to take a look at some commonly abused drugs just to get a better idea of how drugs actually work at the level of the synapse and how they ultimately give the effects that they give and how addiction is achieved through reward pathways, etc. So let's take a look inside here. Alright, so what we can do here is we can grab each one of these mice and go ahead and put them in the chair to take a closer look at what's happening. So why don't we start with our alcohol mouse. And we're going to take a look at what alcohol does to the brain here.
Inhibitory neurotransmitters called GABA are active throughout the brain. These neurotransmitters act to control neural activity along many brain pathways. When GABA binds to its receptor, the cell is less likely to fire.
So that's something we talked about, GABA being the major inhibitory neurotransmitter of the brain, right?
Meanwhile, in another area of the brain, another neurotransmitter called glutamate acts as the brain's general-purpose excitatory neurotransmitter. When alcohol enters the brain, it delivers a double sedative punch. First, it interacts with GABA receptors to make them even more inhibitory.
So remember here, we're enhancing the function of the GABA receptor by binding to an allosteric site. So we talked about this previously, right?
Second, it binds to glutamate receptors, preventing the glutamate from exciting the cell.
So it's not surprising that alcohol is a depressant, right? Because on the one hand, we're enhancing inhibition, and on the other hand, we're blocking excitation. So these are really two different effects that go together.
Alcohol particularly affects areas of the brain involved in memory formation, decision-making, and impulse control.
And again, some of the behavioral effects of drinking alcohol, so not so surprising, right? When we think about the fact that it's affecting the prefrontal cortex area, which is highly involved in decision making and is critical for actually controlling your ability to abstain from doing certain types of things that you know are not appropriate or safe, etc. Alright, so let's go to our methamphetamine mouse. Methamphetamine is something that was discussed in the video lecture, so let's see, just to review sort of what happens here.
Dopamine transporters are responsible for removing dopamine from the synaptic cleft. Because meth mimics dopamine, it is taken into the cell by the dopamine transporters.
So remember, here we have methamphetamine getting into the brain. It's actually pulled in through the dopamine transporter. Now, the dopamine transporter, DAT, is normally supposed to just reuptake dopamine from the synapse back into the presynaptic neuron. But here we're actually taking drug in
Once
instead.
inside the cell, meth enters the dopamine vesicles, forcing the dopamine molecules out.
So the second part, you have entry, then you have it actually entering into vesicles, pushing dopamine out, and now the dopamine is going to get released in larger quantities through the next mechanism.
The excess dopamine in the cell causes the transporters to start working in reverse, actively pumping dopamine out of the cell and into the synapse.
So here you have a transporter that works, but the gradient that's produced by the excess dopamine inside the terminal causes the transporter to actually start kicking dopamine out. So we now have a flood of dopamine entering the synapse.
Excess dopamine becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell. Meth is highly addictive because it works directly on the brain's reward pathway, making the user feel intense pleasure and exhilaration.
And because we're experts now, we know that they're actually talking about the mesolimbic dopamine pathway, from the ventral tegmental area to the nucleus accumbens. Now let's take a look at our MDMA mouse. Now this is what oftentimes we think of as ecstasy or Molly sometimes in its purest form of MDMA.
Serotonin transporters are responsible for removing serotonin molecules from the synaptic cleft after they have done their job.
So again, we're looking at the level of a transporter here, right? This is SERT, so this is the serotonin reuptake transporter.
Ecstasy mimics serotonin and is taken up by serotonin transporters. In fact, ecstasy is more readily taken up than serotonin itself.
So this is similar to what we saw with methamphetamine, right? We have uptake of the drug into the cell. Remember, these things have to get in somehow, right? So it's brought in through SERT, okay, through the serotonin transporter.
This interaction with ecstasy alters the transporter. The transporter becomes temporarily confused and starts to do its job in reverse. The transporter starts transporting serotonin out of the cell.
Now this is very similar, right, to what we saw with methamphetamine, but there is important differences between the effects that are produced from MDMA, or ecstasy, as opposed to methamphetamine. So even though they have similar mechanisms of action, the areas where they act, right, and actually has a profound effect on the behavioral outcome of actually taking one of these drugs.
The excess serotonin becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell. Ecstasy affects serotonin pathways responsible for mood, sleep, perception, and appetite. Ecstasy also indirectly interacts with the reward pathway. The excess serotonin stimulates a milder release of dopamine along the reward pathway, giving ecstasy slightly addictive properties.
So here you can really see the point that we wanted to hammer home when we started talking about pharmacology, and we really wanted to learn a little bit about neural pathways, even though they're very complicated. Essentially, the point here is that depending on where, right, these different targets are in the brain, it's going to have profound effects on function. So you can see here that ecstasy, MDMA, actually acts in a wide variety of different areas. Okay, so it actually affects several different types of functions. So those were compounds or drugs that were touched upon in the video lecture itself, but I want to actually show you the mechanisms of action of a few of these other commonly abused drugs as well. So let's go ahead and let's do cocaine next, because in some ways this is very similar. Now, cocaine, of course, is a stimulant, right? Most people know that.
Dopamine transporters are responsible for removing dopamine molecules from the synaptic cleft after they have done their job.
Alright, so here again we're at the dopaminergic synapse, right? So we have DAT, D-A-T, the dopamine transporter.
Cocaine blocks these transporters, leaving dopamine trapped in the synaptic cleft. As a result, dopamine binds again and again to the receptors, overstimulating the cell.
So again, this is a slightly different mechanism, right, of flooding the synapse, because really what we're doing here is we're not actually reversing a transporter and kicking more neurotransmitter out, but what we're doing is we're keeping the neurotransmitter in the synapse for a longer period of time, right? And so that means that it's going to continue to bind and bind.
Like other drugs, cocaine concentrates in the reward pathway. However, it is also active in the part of the brain controlling voluntary movements. This is why cocaine abusers are fidgety and unable to be still.
So if you'll recall, two of the major dopamine pathways that we discussed were the mesolimbic pathway, which is the reward pathway, and the nigrostriatal pathway, right? So that's the pathway that actually goes from the substantia nigra up to the striatum, okay, and controls movement. So this explains why cocaine abuse results in after effects where you tend to be a little bit jittery, a little bit fidgety, right? Shaky, because what's happening is essentially that motor circuit is being altered. It's being modulated by excessive stimulation by cocaine. Pretty interesting, right? So let's see. I want to save our LSD mouse till the end. Let's see what happens when we smoke marijuana. Or nowadays, take a THC capsule, which is a little bit more popular than it used to be.
Before marijuana enters the system, inhibitory neurotransmitters are active in the synapse. These neurotransmitters inhibit dopamine from being released.
Now, we haven't really talked too much about cannabinoid receptors, okay? But cannabinoid receptors are receptors for endogenous opioids in the body. And these are actually involved in fine-tuning a lot of different processes in our bodies, okay? They're also involved to some degree in pain signaling and different things like that.
When activated by the body's own native cannabinoid, called anandamide, cannabinoid receptors turn off the release of inhibitory neurotransmitters. Without inhibition, dopamine can be released.
So essentially the point here is you have sort of a few different neurons at play here, okay? So you have your dopamine neuron. Now we know what happens when dopamine is released, right? It's going to bind to its receptors and it's going to produce a rewarding effect, right? And euphoria and different things like that. Well, there is a inhibitory system, right? Largely driven by GABAergic neurotransmission, so GABA, right, an inhibitory transmitter, that holds this in check and limits the cell from firing, so it limits dopamine release, okay? Now what happens is when anandamide binds, this is the endogenous ligand for this receptor, it actually blocks the release of the inhibitory transmitter, okay? So it's like disinhibition, right? So normally you would have inhibition here, but because this is bound, now this cannot inhibit this cell anymore. So when this is bound, this guy fires more. All right? Does that make sense?
THC, the active chemical in marijuana, mimics anandamide and binds to cannabinoid receptors. Inhibition is turned off and dopamine is allowed to squirt into the synapse.
So you see what happens here. THC, which is the active ingredient in marijuana, actually or the active component, I should say. We don't typically make this, but it's although you could make it. This is actually binding to the endogenous ligand's binding site, right? The anandamide binding site, and causing more dopamine to be released.
Anandamide is known to be involved in removing unnecessary short-term memories. It is also responsible for slowing down movement, making us feel relaxed and calm. Unlike THC, anandamide breaks down very quickly in the body. That explains why anandamide doesn't produce a perpetual natural high.
So when we think about the after effects and the long-term effects associated with marijuana use, we typically think of things like having trouble with memory, all right, and particularly short-term memories, and perhaps a decrease in motivation, right, which is kind of like a little bit of a slowing down, right, being a little bit more calm and relaxed. So this is actually all of these traits, all of these characteristics are actually connected with the endogenous ligand anandamide, okay, which is actually the same thing, binds to the same site as THC binds to. So this actually explains some of those effects as well, right? All right, we got 2 more to do. I think 2 more. So let's go ahead and do our heroin mouse.
Before heroin enters the system, inhibitory neurotransmitters are active in the synapse. These neurotransmitters inhibit dopamine from being released.
All right, so again, we have a situation here where we have inhibition, okay, so we have neurons that are going to release inhibitory neurotransmitters that are going to block the release of dopamine, okay, and again, we have an opiate receptor over here, so you might be able to guess what you think is going to happen here. Let's see. What is the main characteristic of heroin? How does it actually manifest physically after somebody takes heroin?
When the body's natural opiates activate opiate receptors, the release of inhibitory neurotransmitters is shut down. Without inhibition, dopamine can be released.
Okay, similar story, right?
Heroin mimics natural opiates and binds to opiate receptors, turning off dopamine inhibition. Dopamine is allowed to flood the synapse, producing immediate feelings of sedation and well-being.
So heroin is probably one of the main drugs of abuse that we think of when we think of something that really induces an intense feeling of euphoria, right? So it sort of stands to reason that yes, we're going to have this massive dopamine release into the synapse.
Neurons with opiate receptors are in parts of the brain responsible for the transmission of pain signals, stress response, and emotional attachment. Our body's opiates are natural painkillers, effective when we have sustained massive injury. This is why morphine, a drug related to heroin, is used as a painkiller.
All right, so again, here we see the difference, right? Even though we're acting in a similar mechanism, right? Now we saw with marijuana that we're actually binding to a cannabinoid receptor. But here, okay, what we're actually seeing is that we have many different regions of the brain that contain these types of receptors that this binds to, right? And so what this does is it will affect a whole bunch of different systems, and it will affect a bunch of different types of behavior, okay? And in this case, we see emotional attachment, right? So this is one thing that's a hallmark of somebody who's a heroin user, is they start to detach, right? And this is basically due to the fact that overstimulation of this system, right, tends to lead to an after effect where we're depleting this emotional pathway, these neurotransmitters, and so it becomes difficult for them to connect emotionally. People who use heroin repeatedly. And last but not least, our psychedelic mouse.
LSD acts almost exclusively on serotonin neurons. LSD chemically resembles serotonin and elicits its effect by binding to serotonin receptors.
All right. So this is again, this acts essentially like serotonin. So it's
There are several
binding to
types
these
of serotonin receptors in the brain. Each is responsible for performing specific functions. LSD interacts with particular receptors, but not always in the same way. Sometimes LSD may inhibit them, and sometimes it may excite them. This is one reason why LSD has complex sensory effects. LSD and other hallucinogens excite a particular region of the brain known as the locus coeruleus, or LC. A single neuron from the LC may branch into many different sensory areas of the brain. The LC is responsible for feelings of wakefulness and evoking a startle response to unexpected stimulus.
So you remember when we went through the basic neuroanatomy that the locus coeruleus is actually the center of what? Well, it's the place where we have a lot of adrenergic neurons actually making norepinephrine, right? And we said that norepinephrine was a neurotransmitter that was very important in attention, right? And attending to a new stimulus when it's presented. Okay, so it sort of makes sense if you think of the hallucinogens. What's happening is you're actually perceiving, and your perception gets a little bit distorted, right? You have hallucinations, you're seeing things that aren't there, you're hearing things that aren't really there, because there is actually a potent action on a system that's involved in actually noticing things, right? So this is sort of the biological basis of where that outcome comes from taking LSD. So I hope this gave you some insight on how these different types of drugs act at the synapse, and how even though they might act on similar mechanisms, depending upon where the receptors are located and where the pathways are in the brain, we can have many different types of effects, okay, on the body. So I will see you next time.